Hi, this is Maggie Vires, and this is my Exam 5 presentation on saprobes and the decomposition process. So first I'm going to talk about what saprobes are. So saprobes, they are organisms that help clean up the natural world. So they thrive on and feed off of dead or decaying matter. So think of like leaves that have fallen from a tree, they're decaying, or remains of animals. So without saprobes, this dead waste would pile up, but instead the nutrients are being recycled and thrived off of. So bacteria and fungi, they are both forms of saprobes. They can be both forms of saprobes due to their enzymes. So they can break down the compounds outside of them. And saprobes, they're able to recycle nitrogen and carbon and also put it back into the soil and other things in the environment. So now we're going to talk about how they break down the matter. So the main thing to understand is that saprobes digest things externally, like outside of their body, and then they'll absorb it. So animals, they chew and digest food internally. Well, saprobes, they'll release enzymes directly into the environment. And the enzymes are strong and they can break down large molecules into smaller pieces. And after it's been broken down, the fungi or bacteria, they're able to absorb the nutrients back into their cell walls. And like I said before, not all the nutrients goes back to the decomposer. It also can be put back into the soil and enrich it or other organisms. And this process of digesting the thing and bringing it in, this is called extracellular digestion. So when we think about saprobes, we need to know that enzymes are really what makes them able to do what they do. So they don't ingest food like animals. They excrete the enzymes into their environment and bring in the nutrients. So enzymes like cellulases are used for plant material, or proteases for proteins, lipases for fat. These are all different examples of enzymes, and they break down the polymers into much smaller monomers. And like cellulase, it like chops up the cellulose into glucose. And this goes the same for proteins. So proteases will reduce the proteins into much smaller amino acids. And this is called predigestion. And this happens outside of the microbe in the extracellular space. So this is really why it's called extracellular digestion. And so once the substrates are reduced into something smaller like monomers, the microbe, it needs to pull the nutrients back inside the cell, where there's different specialized transport proteins that help move them in, like the nutrients like glucose, or can be taken up through facilitated diffusion, or ATP channels or proton gradients are used to help move against the concentration gradients. So after they enter, the nutrients will feed into different metabolic processes like glycolysis, like sugars go into glycolysis, or amino acids are used to build proteins, and fatty acids for energy. And saprobes, they are efficient in recycling this organic matter and using it to their advantage, and then eventually returning it back into the ecosystem. So now I'm going to talk about fungi. So we talked about that it can be fungi or bacteria. So this process is very controlled, and it'll enter through, usually for my example, we're going to talk about wood. So it'll enter through a tough material like wood, and it'll sense that the nutrients are low. And it uses surface receptors and internal signals that will activate genes that will make them secrete these enzymes and break down the lignin, cellulose, hemicellulose, and then inside the fungal cell, ribosomes will help manufacture these enzymes. So They're processed and packaged from the ER to the Golgi apparatus and then secreted through the cell wall and into the surrounding wood. And this process, we talk about extracellular digestion, happens outside the cell, but there's different methods that different fungi use. So white rot fungi, these release enzymes and they produce like oxygen reactive species. They break apart the lignin and expose the cellulose underneath. However, brown rot fungi, they use chemical reactions to break down the cellulose and leave the lignin intact, while soft rot fungi, they break down cellulose and lignin and at the same time. So once these enzymes, they've broken down the large polymers into smaller molecules like glucose, amino acids, the fungus will absorb them through the cell wall and into the plasma membrane through transporter proteins. So inside the cell, these nutrients will feed metabolism, growth, and allow the hyphae to expand deeper into the decaying material, which is wood in this section. So the hyphae and the mycelium, these are important for growth and spreading. So as this cycle continues, the mycelium or the mycelial network, it will grow and grow and grow, and it'll keep recycling organic matter and return nutrients back into the soil like carbon and nitrogen. And without this, they wouldn't just continue to grow as rapidly as they do. So now I'm going to talk about bacteria. So bacteria, when it detects a nutrient, it will see that the organic matter is available and that nutrients are low. And so inside, it will trigger signals that turn on the genes and code for the enzymes. So proteases will break down proteins, lipases will break down fats, like that. So cellulases are in charge of plants or fungal debris. So bacteria, they're better than fungi at breaking it down. So they release nitrogen and phosphorus back into the environment, and their nutrients help enrich the soil just like fungi, but they don't usually work alone. They work with fungi. So fungi will attack something hard like the wood that bacteria wouldn't be able to handle, and bacteria will take up whatever's left over in their decomposition process and helps also recycle that stuff. So bacterial decomposition, it begins with the sensing. So when they're in close contact with the dead material or decaying, like the leaf or whatever, they'll have chemical signals that will sense in their surroundings. They'll sense changes in concentration, and when that happens, internally, it'll signal for genes to turn on and the enzymes to secrete. So it'll secrete cellulases and for fungal debris. So once the enzyme is produced and exported across the plasma membrane and the cell wall, the enzymes will turn the large organic molecules into shares like acids into smaller pieces. The molecules are then absorbed back in, and transporter proteins will put them back into the membrane and use it for cellular respiration or biosynthesis. So during this, bacteria will release small inorganic molecules like carbon dioxide and phosphate and ammonium back into the environment. And that's all for my slides. Thank you.